All right, so cellular transport. We're going to start off with our levels of organization. So we start technically with atom, molecule, and then organelle, but we're really going to start with the cell for this class. And then the cell to the tissue, tissues to organs, organ to organ systems, and organ systems to organism. So here we have that, the same thing, just in words. So you start with a cell. Cells make up tissue, and tissue is a group of cells that perform the same function. So lung tissue exchanges gases. It has a specific function. And then you put that tissue together and you make up an organ. And that would be like our lung. And so it's made from the specific tissue that is unique to what it needs to do. And then you put organs together and you get an organ system. And so our lung would fit into our respiratory system. And then you put all of the organ systems together and you get your organism. So you as a whole are an organism made up of organ systems, which are made up of organs, which are made up of tissues, which are made up of cells. And cells are our basic unit that we have talked about. So that is the smallest that we will go. We start with cell, that's the most simple, and then we get to the most complex down here with organism. Okay, our plasma membrane or cell membrane, those mean the same thing. And you will need to know how to identify these different components in on a diagram. So make sure you know what these look like and also what they do, which is what we're about to talk about. Okay, so parts of the plasma membrane. Our first part is a phospholipid. Phospholipids are a bilayer, and we've talked about how bi means two. So it's a two-layer part, and it's the majority of the membrane. So when we look back at our picture here, all these little purple heads and these little tails, all the way across are phospholipids, so the majority of the membrane. And this is going to regulate molecules that enter and exit the cell. So this is going to kind of say like, you can come through easy, you can't, you get to leave, all that kind of stuff. And so we're going to talk about what each of these parts is. The bilayer means that there are two layers of this here. And we know that hydro means water, and we know that philia means love, and we know that phobia means fear or dislike. So hydrophilic means that this head right here, the phosphate group at the top, loves water. And we know based on our words here that hydrophobic, our lipid tail, does not like water. It dislikes water or fears water. So one side is hydrophobic and one side is hydrophilic. The next part of the plasma membrane is our membrane proteins. These are going to provide support and strength. And if you think about when people go to the gym and work out to get strong, you know, they try to eat a lot of protein. So that might be a good way to help you remember that this adds some strength to our plasma membrane. They also are going to be responsible for helping transport certain molecules across the membrane. Some molecules can't just slide through the phospholipids, so they're going to need help. And these proteins are going to be there to help them cross. And we'll talk more about that when we get to passive transport in a little bit. Then we have our carbohydrate chain. This is like the little walkie-talkie, and it looks like this. This is going to be for cell-to-cell communication or a receptor. So this is how the cells are going to communicate with each other. And then cholesterol. I think when everyone thinks about cholesterol, it's like that weird, like cholesterol is bad. But in reality, you have to have cholesterol in your body, and it's going to increase the flexibility or fluidity of the membrane, which is super important. Is a super important part of our membrane. So cholesterol increases flexibility. The cell membrane, the plasma membrane, is called the fluid mosaic model because we have all these different parts. They're able to move around, so it's fluid, and it's mosaic, which means made up of all the different components. Now we're going to talk about active and passive transport. So you can see here, this is passive, this is active. The main difference between these two is one uses energy and one does not. Passive is not going to use any ATP or energy, and active, you can see energy is being put in to the process. So active is going to use energy. So let's start with passive. So passive transport, no energy is used. The molecule movement is going from high to low concentration down the concentration gradient. So let's talk about what that all means. So high concentration, let's think of this as two rooms side by side. So one room is really full of people. It's almost packed full. And then the other room, there's like 3 people chilling in there. So passive, they're like, they're not a whole lot of energy involved here. Actually, no energy. I don't want to confuse you. So no energy. So these people, they don't feel like fighting with all these other people for space. They're just going to walk next door to the other room. And there's going to be space for them. It didn't take a lot of energy for them to get to that other room. When you think of down the concentration gradient, you think we go from high. So if you high up here to low down, so you're going to be down the concentration gradient. And it says think flowing downstream. So it's easy. It's just happening. Not a whole lot has to happen. This is just happening based on the concentrations that are in each, in the outside of the cell and in the inside of the cell. Cells really want to be in equilibrium, sorry. So they are going to constantly try to maintain, okay, there's 5 people in this room, there needs to be 5 people in this room. So they're going to try to go back and forth based on where they would go to make it equal. So that's how passive transport is decided when they're like, that's how they decide when to move. Molecules are always moving across the membrane while maintaining equilibrium. So this kind of is there to make you know that it's not just like, okay, now we have 5 people in each room, we're good to go and we're done. They're always going to kind of be moving back and forth. There are 3 types that we're going to talk about: diffusion, osmosis, and facilitated diffusion. So diffusion is the random movement of molecules, no membrane necessary. So if you look at our picture over there, you put a lump of sugar into water, and over time, the sugar is going to diffuse throughout all of the water. There's not an order. It's kind of just random movement of these sugar molecules throughout the water. Osmosis is that same process that we see with diffusion, but this is going to just be talking about water, the movement of water back and forth through the membrane. Then facilitated diffusion, molecules move across with the help of a channel or carrier protein. So I told you these proteins were going to come back into play that we talked about being part of the plasma membrane. And so this is where they come in handy during facilitated diffusion. So some molecules like glucose wouldn't be able just to go through the phospholipids. So they are going to use these proteins that help them get to the other side of the membrane. And I don't want you to get confused, because even though it looks like something is having to happen and it's trying to help and all of that, this is still part of passive transport. So no energy is involved. Now we're going to talk about our types of solution. So we have an isotonic, hypertonic, and hypotonic. Isotonic, I think, is the easiest to remember because it's the same concentration on the inside and the outside. There's no back and forth or wait, which one was that? So isotonic is equal, same concentration inside and outside of the cell. Hypertonic is easy to remember if you think of high, hypertonic. So it sounds like the word high, and the solution has a higher concentration. So if we're talking about the hypertonic solution, the solution would have a higher concentration than the inside of the cell. So hypotonic is the opposite of that, and hypo rhymes with low. So it's easy to remember that hypotonic is a solution with lower concentration than is on the inside of the cell. Okay, and then we have, the reason that this is important to our bodies is because it affects the size of our cells when they're placed in a hypotonic solution. So we're going to talk about what that does to the cell. Isotonic, again, super easy. The cell stays the same size. Hypertonic, the hyper, so this solution is more concentrated than the inside of the cell. Water is always going to move toward where there is more concentration. So water in this case moves out of the cell, and it makes sense that when water leaves the cell is going to shrink or shrivel. And I'll come back to the plant stuff in just a second. So with hypotonic, the higher concentration is inside of the cell. So water is going to move into the cell. Hypotonic, the solution is lower. So water wants to move to where there's more concentration. It's going to move to the inside of the cell. And eventually the cell is going to swell and swell, right? And if you think about like a water balloon, as the cell swells, like if you were putting water into the balloon, it's going to get bigger and bigger and bigger, and eventually it's going to burst. So same thing happens to animal cells. The reason that that the plant cell will not burst is because they have a cell wall. So while the cell membrane inside of the cell wall will expand and expand, it can only get as big as the cell wall. And so if we went back to our hypertonic solution, plant cells, again, have the cell wall. So they're still going to have that basic structure, the same size, but that cell membrane on the inside is going to pull away from that wall and get smaller and smaller. So if we look here, I'll have to go into our slideshow. So if we get here and we see that we put a plant cell in distilled water, this is just plain water. We've taken out all of the concentration, and so it would be at 0. And we know that when it's placed in a hypotonic solution, water wants to go to where it's more concentrated. So it's going to go into the cell. If this was an animal cell, it bursts. But since we're looking at plant cells, it just pushes itself, the cell membrane, up against the cell wall, and it stiffens and keeps its shape. If we put it in a concentrated salt solution, it is going to shrivel up and shrink because this is a hypertonic solution. Water wants to move to where it's more concentrated. So water is going to move out of the cell, and the cell membrane is going to shrink and pull away from the cell wall. Then we have animal cells. So same thing, distilled water, hypotonic solution. The concentration in the cell is higher than it is in the water. So water wants to go into our cell. In this case, the cell is going to swell and eventually it's going to burst. If we put it in an animal cell in a concentrated salt solution, water always wants to go to where there's more concentration. So it's going to go out of the cell this time, and the cell is going to shrink and shrivel. All right, back to our active and passive transport. We're moving on to active now. So active is pretty much the opposite of passive, not the opposite in the sense that they're both going to move things across the membrane. One is just going to use energy. And so active transport uses energy. These molecules are going to go from low concentration to high concentration. So let's go back to our 2 classrooms side by side. Now you're in the room that only has 3 people in it. The other room has 150. It is like crazy crammed full, and it's going to take a lot of energy for you to get back into that room because there's hardly any space. So you are going to push and push and push your way back into that room. It takes energy. So you're going from a place there, it's low concentration to high concentration, and it takes energy to do that. This is going to be up the concentration gradient. So if you think about being low and then going to high, that is up the concentration gradient. And think of swimming upstream. That just means think about how much more difficult it would be to swim upstream. It's going to take energy. The sodium potassium pump is one of my favorite things. I don't know why. I've just always loved this ever since college. I just think it's really cool. So this process consumes about half of a cell's energy. So clearly it's very important. And it's essential in the cell membranes of muscle and nerve cells. So the sodium potassium pump is responsible for allowing our muscles to contract and for nerve cells to be able to do what they do. So when you get a cramp, if you've ever like had a cramp from running or playing a sport or whatever it is, and you think about the foods that they tell you to eat, you're probably going to come up with like a banana or mustard or pickles or something like that. And the reason for those foods is that we want to eat potassium, which you see in here, and sodium, which is part of salt, right? So those foods either have salt or potassium. And that's because of this sodium potassium pump. If these levels get off, or if there's no potassium, if there's too little or no sodium or too little sodium, it's going to cause problems in the contraction of our muscles and in our neurons. So think about if you get a cramp, your muscles have stopped like releasing that contraction. So they have held that contraction. And if you've had a cramp, you know exactly what I mean, but your muscles are contracted and they aren't letting go. So that's one thing that could happen if the sodium potassium pump is off balance, or the opposite of where your muscles have no ability to contract anymore. And so obviously we need our muscles and our neurons to do what we do every day and to survive. So it's important that this sodium potassium pump in your cells is working. Okay, so two types of active transport, bulk active transport. We have endocytosis and exocytosis. Endocytosis is engulfing of particles via a vesicle. So you can see down here, we have a start of how it's engulfing, and then you see it and it kind of breaks off into a circle, and that's the vesicle. And we have two types of this. One is for food and one is for liquids. Phagocytosis is for food. Phago, food, they start with the same sound. It might help you remember. And then pinocytosis, it engulfs liquids. So phago and pinocytosis they are types of endocytosis, and endocytosis is our first type of bulk active transport. Our second type of active transport is exocytosis, and this is easy to remember because exo, exit. So endocytosis is bringing things into the cell, and exocytosis is pushing things out of the cell, exiting the cell. Sorry, the bell is ringing because I'm at school. And this is going to be removing, exiting of waste or particles via a vesicle. So you can see here, it pretty much looks the same except for its opposite. So now we start with our little vesicle that's detached, and it kind of gets closer to the membrane and then eventually releases those particles. All right, so that is all of our notes for this unit. Please email me if you have any questions, and I hope you have a wonderful day.